While we often view aging as the passive passing of time, your cells are engaged in a high-volume, physical struggle for integrity. At the microscopic level, aging is the invisible accumulation of uncorrected errors inside your cells. Every single day, the DNA within just one of your cells sustains tens of thousands of individual damaging events. These strikes come from two directions. You face internal exhaust from your own metabolism, like reactive oxygen species, and external strikes from the outside environment. The only reason you don't instantly dissolve under this heavy assault is because you possess a highly advanced, built-in biological operating system designed exclusively to manage and execute cellular repair. As we get older, this control system doesn't disappear or break, it simply gets silenced. The question is, what if we could turn it back on? At the center of this defense network is a highly conserved family of signaling proteins called sirtuins. Specifically, Sirt1 and Sirt6 act as the supervisors of your cellular repair crews. But these supervisors face a physical obstacle. Your DNA is tightly spooled and locked away inside protective vaults called chromatin. This keeps the genetic code safe, but it makes damaged sequences completely inaccessible to the repair enzymes trying to fix them. Sirtuins solve this problem by rushing to the site of the damage. They perform a chemical reaction known as deacetylation, which forces the protective vault to dynamically unspool. This opens up the chromatin and allows the repair crews direct access to the fracture. Once the sequence is fixed, sirtuins immediately relock the vault. They recondense the chromatin to secure the genome, which prevents the cell from accidentally reading and replicating a damaged gene. Without sirtuins actively managing this lock-and-key interface, DNA repair stalls. The uncorrected damage accumulates, and the cell eventually enters a state of permanent arrest, known as cellular senescence. There is a severe bottleneck in this system. Sirtuins cannot function on their own. They require a specific cellular coenzyme called NAD+ to fuel their repair protocols. This chart shows how chronological age tracks directly against NAD+ availability. Between the ages of 20 and 50, cellular NAD+ levels drop by roughly half. As the fuel supply plummets, sirtuin activity flatlines, forcing your repair supervisors into a dormant state. Chronological age is only part of the equation, but modern lifestyle habits actively accelerate this rapid biological bankruptcy. The timing of your meals, specifically late-night eating, creates massive friction against your body's natural circadian rhythms. Consuming calories after the sun sets spikes your blood sugar right as melatonin levels are naturally rising. This chemical collision plunges the body into an active anabolic storage state, precisely when it should be shifting into a resting repair state. This severe circadian misalignment effectively blocks the exact metabolic signals required to trigger sirtuin activation. Consuming calories late at night silences the specific metabolic software required to repair your genome while you sleep. We do have a biological workaround to reactivate these dormant sirtuins. Certain plant compounds called polyphenols, such as resveratrol, fisetin, and quercetin, can interface directly with our cellular machinery. This diagram shows a polyphenol molecule binding to a dormant sirtuin enzyme. This physical interaction triggers the protein to activate its repair protocols, even in a compromised cellular environment. These compounds trick the body into a state of metabolic efficiency. They mimic the cellular benefits of fasting, effectively rebooting your genetic repair system without requiring you to endure actual starvation. Aging represents a progressive, preventable loss of control over the exact cellular mechanisms that maintain our genetic stability. Genetic optimization is not about rewriting or altering your base DNA code. Optimization means consciously influencing how your existing genes respond, repair, and maintain themselves over a lifetime through your specific environmental inputs. You maintain this system by prioritizing nutrient-dense foods, leveraging polyphenol-rich plants, and practicing circadian fasting to protect and preserve your NAD+ levels. Every single day, through the exact timing and quality of your inputs, you are either actively funding your body's repair system or you are allowing the microscopic damage to compound. To learn more actionable, science-based strategies on cellular longevity, metabolic alignment, and living with intention, subscribe to the channel. We often view aging as a progression of time, but on a cellular level, it represents the residue of accumulating unrepaired damage. Every day, the simple mechanics of metabolism and external stress inflict tens of thousands of molecular breaks within the DNA of every single cell. To counter this, our bodies rely on a family of specialized proteins called sirtuins, enzymes that detect these breaks and coordinate the cellular response. These proteins don't operate in a vacuum. They wait for specific biochemical signals to move the body out of a passive survival state and into a focused repair cycle. By identifying and optimizing these signals, we can influence the rate at which our cells manage the damage that drives biological aging. DNA is rarely found in a straight line. It is wound tightly around histone proteins. This configuration is efficient for storage, but it physically blocks the cellular machinery from reaching a damaged sequence. When the cell detects a double-strand break, Sirt6 and Sirt1 are among the first to arrive at the site, docking directly onto the histone clusters. They perform a process called deacetylation, which causes the histone coils to relax. As the DNA unspools, the repair enzymes finally have the physical space they need to bridge the gap and mend the strand. Once the sequence is restored, the sirtuins work to recondense the chromatin, winding the DNA back into its original, secure structure to prevent the cell from misreading the newly repaired code. This constant cycle of unspooling and respooling is a primary mechanism that prevents daily genetic wear and tear from becoming permanent. Even with these enzymes in place, the system is fragile. Sirtuins require a specific coenzyme called NAD+ to power their activity. This chart highlights a significant bottleneck in human biology. By age 50, our natural supply of NAD+ has typically dropped by half, limiting the resources available for these repair pathways. When the fuel supply is low, sirtuins cannot efficiently open the chromatin. This leaves the most dangerous double-strand breaks to accumulate within the genome. Scientists visualize this backlog using a COMET assay. In this image, the long, smeared tails trailing behind the cellular nuclei are actually physical fragments of broken DNA. This accumulation is known as oxidative debt. It provides physical evidence that the aging process is tied to a breakdown in our internal maintenance supply chain. If aging is driven by this lack of fuel and activation, the focus shifts to how we can manually restore the environment necessary for repair. Specific plant compounds, such as resveratrol, have been shown to bind to the Sirt1 enzyme, mimicking the signals that trigger its repair functions. Research into nutrient synergy suggests that combining these activators with NAD+ precursors can help re-energize the system, providing both the signal to begin repairs and the fuel to finish them. While these molecular interventions are powerful, their effectiveness depends on whether we provide the cell with a stable environment to perform its maintenance. This environment is governed largely by the timing of our metabolic signals, specifically when we choose to stop eating. Eating late into the night triggers blood sugar spikes and metabolic shifts that force our cells to remain in a high-intensity processing state when they should be transitioning into rest. This diagram illustrates the resource trade-off. Digestion is metabolically expensive. It diverts the cell's limited pool of NAD+ away from DNA repair to manage the immediate demands of processing nutrients instead. Ending food intake 3 hours before sleep helps provide the quiet window necessary for these sirtuin-led pathways to focus entirely on genomic maintenance. We are beginning to see that aging is not an unalterable slide toward decline. By providing the body with the right molecular fuel and respecting its metabolic timing, we can support the pathways that preserve our genomic integrity. We often imagine aging as a car rusting in a field, a slow, inevitable decay caused by the simple passage of time. But at the microscopic level, the reality is a constant struggle. Our cellular blueprint faces a continuous bombardment of oxidative stress, leading to severe physical fractures known as DNA double-strand breaks. Human cells possess highly sophisticated machinery specifically designed to fix these fractures. Strangely, as the years pass, this built-in repair crew seems to clock out and stop working. Our internal tools remain present as we age, but the instruction network that tells those tools when and where to work begins to falter. Human biology operates on circadian rhythms, internal clocks programmed to process fuel during the light hours and prioritize tissue repair in the dark. Late-night eating forces a direct conflict with this schedule. This data illustrates the metabolic cost of that conflict. This graph compares blood sugar responses, a massive, prolonged spike for a meal eaten at 10 p.m. versus a much smaller, quickly resolving curve for the exact same food consumed at 6 p.m. By forcing the system to process fuel late into the night, we create a biological environment where the rate of cellular damage begins to outpace the speed of our natural repair mechanisms. To understand how to reverse that pace, we have to look at the master controllers of the cellular repair crew, a family of enzymes known as sirtuins, specifically Sirt1 and Sirt6. These enzymes solve a difficult physical problem. In a healthy state, our DNA is wound incredibly tight around proteins called histones. To reach a broken strand, sirtuins must physically unspool this dense chromatin structure so the repair machinery can access the sequence. Once the sequence is patched, sirtuins must immediately recondense the strand. Leaving it unspooled is dangerous, as it can trigger the accidental reading of damaged genes. These sirtuins are incredibly powerful, yet they sit completely inert by default. They require highly specific environmental signals to turn on and do their jobs. The first required signal is energy stress. During periods of fasting or caloric restriction, a nutrient sensor inside the cell called AMPK detects the drop in energy levels. AMPK acts as a master cellular switch. It forces the body to move out of its default state of growth and storage and into a state of maintenance and repair. This shift leads us to NAD+, the mandatory physical fuel sirtuins must consume to perform every chemical reaction. This chart tracks human NAD+ concentration. Between our 20s and 50s, this critical fuel drops by approximately 50%. Strategic fasting to create energy stress, combined with the restoration of NAD+ fuel, forms the non-negotiable foundation of the cellular repair network. The third signal involves plant compounds called polyphenols, like resveratrol. These function as cellular messengers, modulating the repair environment from the outside. Polyphenols bind directly to the sirtuin enzyme, physically altering its shape. This adjustment makes the enzyme more efficient, allowing it to activate even when the available fuel levels are low. The fourth signal is low inflammation. Systemic inflammation acts as biological static, drowning out repair commands. Overwhelmed by an oxidative burden, sirtuin sensors are distracted. They abandon routine DNA repair to fight acute cellular fires, leaving our blueprint to decay. Energy stress and NAD+ provide power. Low inflammation and polyphenols clear static, ensuring the repair crew stays focused. This interconnected network is why taking a single supplement or isolated longevity pill usually fails to show results. Our biology requires the synergy of the entire signal network to trigger a meaningful repair response. Aligning these signals requires a combined approach, stopping food intake 3 hours before bed to respect circadian rhythms, using fasting to activate AMPK, and introducing specific nutrient combinations. When these 4 variables align, the cell enters a synchronized, highly efficient state of continuous DNA repair and epigenetic stabilization. By mastering these metabolic signals, we transform aging from an inevitable decline into a lifelong practice of manageable cellular maintenance.